I'm happy to share what's happening here in Tennessee. So, am I sharing the right one, or?
We just lost your slides.
Oh, okay, okay, all right.
Got it.
Okay, all right. I like to introduce some of our new members here. Many of you know Brand England, who is our field research coordinator. He take care of all the rose plant planting and taking care of all how to keep them alive. And so then from this fall, Elena Pokhrel, a PhD student, joined to our lab, and she will be mostly focusing on the mite population study. Then Deepika Sharma, as a master student, she joined from also from this fall, and she will help us doing this qPCR for all the leaves we collected this year. And many of you know Sarah Collins. It's a little bit sad news here. Sarah left us from this summer, and she is joining the fire academy. Maybe within a few years we'll see she is a firefighter in Tennessee. So, all right, I'll give you a little bit virtual tour of our trials in Crossville, Tennessee. So, many of you have visited these RRD cultivar trials and genetic study that plant these rose plants planted from 2001 to 2023 here. Then this year we planted a GWAS study here in the west side of this world trial. And then in between this trial, we have a mite population study of these 5 blocks here. Then coming towards the center, we have a miticide trials here. And here we have an extension demonstration plot where the general public can go and rate our roses. That's the data what Joe was talking about in the morning. Okay, in the GWAS experiment, we planted around 720 28 plants that Dave also mentioned with the different rows A and B, and each cultivar has 2 replication there. And after planting, then we inoculate these rose plants with the infected rose branches. So I really thankful that crew helped me to inoculate all these roses in the same day. So all these genetic study, cultivar trials, and GWAS study, the sampling was done, and all the roses, all the cultivars, we rated them. The incidence and severity of the RRD was recorded. We have these many rose plants, and we have now more than 3,000 rose leaves we collected. Then we will run qPCR in all of them during this winter. And in GWAS study, as we started this year, I already see some of the symptoms in some cultivars. So it is, and the good thing in GWAS study, only one plant died and all of them survived. So we are really happy on that. So now Bode will talk about some genetic study. Bode,
Yeah.
go from here.
So quickly, what I will be presenting is mostly the genotyping by sequencing. And here we're showing using reads obtained from like an OMSIQ quantitative reduced representation sequencing. We mapped the reads to chinensis, and you can see the chromosome 1 through to 7, as well as the chloroplast and mitochondrial genome. There were also contigs that were not assembled into the pseudomolecules. We also included that in the variant calling, and you can see we have low numbers of markers on that. This diagram here, oh, Jordan.
first one.
Can you go back one slide? Yeah.
Yeah.
So this diagram here at the top just shows the marker density across the genome. So we're showing here that we have a good coverage across the whole genome and good density. In this case, we're reporting under 32,000 SNPs and indels. Advance. Here we're showing the alignment summaries for each of the samples we processed, and the maximum number of reads we aligned to the reference was 98.7%, with the least being 86.4%. So on average, we were able to map 96% of the reads back to the reference genome, and we're expecting that about 4% of the reads that don't map are probably metagenomic in nature or microbial in nature. And we also did an analysis where we determined the proportion of the genome we're actually sequencing. And that came to, on average, 0.23%. Advance. And I want to quickly mention that I've not really provided some backgrounds to what we sampled, but I'll do that when we get to the metagenome section of the project. Here we're showing the proportion of genotypes. Most of them were monomorphic for the reference allele, and about almost 11% were heterozygote. Here we're showing the minor allele frequency, which is a bit high, which is expected in biparental populations. And then here at the bottom, we're showing the mean read depth and the median read depth. Next slide.
That's it.
Okay, that's it. Sorry. Yeah.
Okay. Yeah, then I will talk more about the mite population study experiment. So we have 12 different cultivars. We randomly assign them in 5 different blocks. Then we have 3 replication of each cultivars in each block. So Elena Pokhrel is leading this project. So we planted that in May, and then Elena joined in the fall. Then she has not that much background on mite and viruses. So then I suggest them to visit our collaborators, John and Ron in USDA Belfish Lab. I really like to thank them because they host them for 2 days, and then Elena and Deepika learn about the mites and a lot other information from John and Ron Lab. So then Elena came back, and we samples mainly the flower buds, and at that time sometimes some roses does not have flower buds and flowers, so we also sample the leaf petiole to extract the mite. So we are following the established protocol by Dr. Mark Windham here in our lab, and we extract that mite and we'll do the counting. So Elena has done with the extraction. Now she has already started counting them. So maybe in next update we'll have some data to show about the mite population in different cultivars. So talking about the miticide trials here, I established this trial this year, and we have 6 different treatments, and these are randomly assigned in 5 blocks with 3 replication per block. So we started this fall. So mainly the evaluation, we are thinking to evaluate each cultivar by showing the RRD symptoms. And this also miticide trial we will replicate by other extension group in different states like Georgia, Ohio, and Texas. And we have a regular meeting how we do this, and what are the what evaluation method we are using. I'm also thinking about doing qPCR or mite count population. Okay, so this one is the extension demonstration plot that Joe was talking about. We have this QR code, and expert and the general public can visit in these plots and rate the rose, roseite, and black spot. So in the fall field day in Crossville, so I have attended in between the rose plots, and many people are visited there, and if some interested people participate in our research. The October 10, the Joe talking about, I organize a master gardener training day, and then that day many people show up, and after training, and they really participate on rating these rose, roseite, and black spot fungus, so they are really enthusiastic master garden, and they really care about the research, and they really want to participate. That's why these many people participate in a single day. So with that, I'd like to acknowledge all the funding agency, and definitely Mark Wyndham and Sarah and all the crew. Yeah, thank you. If you have any question, please let me know.
Thank you, Nar and Bodhi. Any questions for
I have a couple of.
Go ahead.
Nar,
Yeah,
did you once you extract the mites,
yeah.
are they still good enough to be, for example, in a liquid or in a suspension, be inoculated to other roses? This is one question.
Oh, okay. So I think we are storing them in a 70% alcohol. Like the extraction materials is the 70% alcohol, so they are not definitely alive on the inoculation. So they are good to count, but not the inoculation.
Yeah. Okay. Good. Yeah. Oh no. Well, this is it. This is it.
Okay. Thank you.
Any other questions for Nar or Bodhi? From anyone?
Yeah, I have a question, Oscar.
Go ahead, please.
Okay.
Yeah, I want to ask, you have mentioned in your PPT regarding the QRT-PCR you will do regarding, so on what criteria you will select, what kind of genes you will go for the expression check via QRT-PCR?
So we are using that RTQ-PCR just for the diagnostic purpose. So we have established protocol developed from Texas A&M. We are using that for the diagnostic of rose roseite virus, and then we are recording the CT values of that qPCR.